So afterwards, if you're interested in watching this over and over again, you can totally do that. You can make it an important part of your life. What am I doing? Cool. So if you're interested in hearing about bcbio and running things, you're in the right place. If you're not, you got to get out real quick. Ready to lock the doors, all that. And so my name is Brad. I'm one of the bcbio developers. Lorena is also here, and she works with me at the Harvard Chan School in beautiful Boston. And yeah, and I think the goal of this is really just there's a cover. I'll talk about kind of a wide range of things. Hopefully it'll be useful, and try to cover everything from like techie, how to run stuff, to like, hey, this is variant calling, and this is some of the things that bcbio does for variant calling, and sort of motivation and why bcbio and all that stuff. So hopefully I cover things that are useful to you. I don't want to talk for 2.5 hours the whole time, so please interrupt me and have discussions. That would be awesome. And if you want these slides, they're online right now. You can go to j.mp, like jump bcbio links, and at the top, that's a link to all the presentations, and these slides are there. And then, as I mentioned, I'm also recording this, so I'll put, what? Yeah, you can talk. Of course, you can talk. This is open source community. You can talk about anything you want. Mean things are fine. Anything is fine. But yeah, I'll record this, so it'll be available there as well. So if you miss any very important things, they're there. Or if you want to point your friends at something, this will be the awesome talk. And so this is kind of the broad outline of what I hope to talk about. What I wanted to kind of do at the beginning was motivate for like what bcbio is and why I think it's worth learning and using, and sort of motivate around community development and why it's hard to build analysis pipelines. And then I'm going to talk specifically about some of the new things in bcbio that make it a lot more interoperable, which are really relevant to this conference as a whole, which is working with the common workflow language. And then I'll talk through some practical examples of how bcbio uses common workflow language to run in multiple places. So on your HPC system, on the cloud, and getting access to Auto 7 bridges. So like sort of demonstrating the usefulness of CWL. So if you're here to learn about common workflow language, this is more of like the, hey, if you use common workflow language, these are cool things you can do. And then I think Michael's talking in the afternoon more like, hey, this is exactly what CWL is. But bcbio is more, hey, we're using CWL, but you never have to sort of know that that's happening underneath. And so then I'll try to talk through some biology, sort of an example from the Personal Genome Project, just an n=1 analysis example, which is hopefully meant to be bcbio does variant calling. Sort of, if you haven't done a lot of variant calling, this is what variant calling is. This is the types of analysis we can do, and sort of walk through those as an example, and then sort of wrap up with lead into. I have the lightning talk about bcbio tomorrow at Bosk, and sort of leading into important things I think about validation and testing we do with bcbio. So, but yeah, please do interrupt me. And these are what I hope you'll take away at the end. So I think the most important thing is science is about collaboration and reuse, and what we're trying to do with bcbio is make that happen using interoperable workflow abstractions, so the common workflow language. I hope you have some idea of how to run bcbio analyses, where you want them to go, or at least pointers to where you would start to get doing that, how to look at and interpret variant calling results, and just to be hopefully motivated and talk about how we can build better things together. So to motivate the whole thing, this is kind of, bcbio does a lot of different things. Lorena's talking, I know, at the conference about the small RNA-seq. It does RNA-seq, it does ChIP-seq. What I'll talk a lot about today is variant calling. It's essentially a set of workflows that call variants in lots of different ways. And so the motivation is you are doing some project, right? And you need to call variants in some way. And so you want to build a variant calling pipeline. So a lot of the places that people go first places to the Bro GAPK, and they look at their best practices. And There's a whole set of things that you can run from mapping to alignment to variant calling, and they walk you through everything that can happen. And now they have these workflow definition language WDL scripts that you can use to run them. So you want to set that up and run it and make it happen. But you want to do it in a way that you want to do more science and less making packages install and making things work. So this is sort of an energy diagram of your effort in, and it's energy, but really effort really in, and your effort and what you're getting out. And you want to get out a lot of science and not a lot of loss in terms of installing a package or trying to make things work or do other non-sciencey things. And so, but there's a ton of barriers if you want to do this yourself and set it up. It starts to look easy at the beginning, and then you get into these issues where you have so many tools that you need to put together and operate. So you want them all to work together. You have to install them. You have to then, once you built it, support it. So once you put it together, then someone's going to say, hey, that's cool. You call variants on human. Can you call them on my dog genome? Can you call them on this rat genome? So you're going to start supporting things. You want to start, then people are going to start, if what you put together is good, then people want to say, hey, it runs on the HPC, but we don't want to use cycles, the HPC is down. Could we run on Amazon, And so you're running on multiple platforms. And hopefully once you're running it, then people will ask important questions like, hey, are you sure it works right? Is everything good? There's a new version of GTK. Is it better or worse? So you need to run validation. So there's a bunch of sort of hidden costs that you don't realize when you just start and you write a shell script to make stuff happen. And so the way that we, what's happening today? Good. Good? We're good, thank you. Sort of the reason we're here, right, is because we believe that we can overcome all these sorts of barriers by working together as a community. And so this conference is sort of that community, right? It's people who want to build things, care about building things, but want to stop building them by themselves and want to work on them together. And so, if you're going, I hope you signed up. There's collaboration sessions after the conference. So this is training, there's 2 days of conference, and then there's up to 4 days of collaboration and working together. And that's, to me, what this conference is really like. This is, helping have a longer form way to talk about things. The conference is a way to learn about everything that's happening. And then, but the real goal is then we all are working together. We're learning how to work together. We're meeting everyone. We're learning who everyone is. And so I hope that the outcome of this conference is building this collaboration. And so what bcbio is that sort of collaboration as a project, right? So this is sort of the high level overview. I call it like a white box software, a clear box software, in that it's meant to be pretty high level, that you take in a configuration file and a set of samples and you run analysis. It does all the work of putting the tools together, scaling them, setting the right parameters, doing the best thing possible for your analysis. And then on the other side, you get out things like variations. If you're doing variant calling, you get RNA-seq. If you're doing that, you get quality results and links into downstream tools. So it's doing all the analysis and running. But the advantage is that, oh, go ahead. Yes. Thanks.
What exactly is a configuration file?
Yeah, so there's like I'll show an example later when we start walking through it, but it's a file in YAML syntax, so that it really defines sort of a high level the type of things you want to do. So it'll say, like, I want to align with BWA, I want to do variant calling with GATK, things like that. So it's meant to be knobs that you can tweak, but sort of bigger knobs than like, this is the parameter I want to run GHK with, or things like that. And so yeah, I'll have some, when I walk through the examples, I'll have some practical so you can see what the syntax looks like as well. So yeah, it's meant to be a higher level of abstraction. So rather than you having to do all of the work together, us as a community come together and decide and figure out a set of pipelines that work well. We work on finding edge cases where they don't work and fix those, sort of make everything work. And the advantage is it's completely open source. You can go in and tweak it. We encourage people to come in and tweak it and contribute. The way it improves is lots of people using it, finding it works well or not well in their data, and suggesting ways to improve it. And so that's kind of the big broad overview, and just, make it practical if you want to run things. These are there's a whole set of documentation about all the different pipelines. It does everything from if you're doing variant calling, germline calling, from single samples to populations. There's cancer calling, structure variant calling. We do RNA-seq, single cell RNA-seq, and small RNA-seq. Over here is representing, and ChIP-seq. So a whole bunch of different things that we're trying to collect together and run and build up best practices for. And at this point, it's normally, hey, you made a pipeline, so what, right? Here's a bunch of things that run, and there's quite a few of these, lots of things that run. And this is you're not meant to read sort of a description from the bpipe software about sort of they built an analysis pipeline and then describing why they built that, as opposed to and you read through many, many different other existing pipelines that exist, because people always come up with excuse to build a new pipeline. There's always some good reason, right? And so the point is there's lots of different choices. What makes bcbio unique or different, or why should you invest time in this versus anything else? And I think the number one reason is sustainability, in that it's a work to build a pipeline, but then the real work is then in maintaining that pipeline over a long period of time. Continue to improve it, iterate on it, supporting people who want to use it, all those sorts of things. And that's kind of been our focus. And so this is a great quote from quite a few years now, I guess now, from Software Carpentry, which I think is a good definition of what makes a software sustainable. It's being sustained if people are using it, fixing it, and improving it rather than replacing it. So the goal is really to build a set of workflows that are sustainable and that people want to build and fix. And the reason for this is that things are changing continuously all the time. So this is I'm always good to update this slide a million times, because it demonstrates both changes and how long bcbio has been going on for. So back when bcbio was a thing at the beginning, it was literally what I was talking about. Is there were really no, if you imagine, there's no pipelines for doing GHK best practice calling, and it was whole genome version 1, and someone wanted to do it. I was working the core, and they wanted to call variants, and so I said I can put something together that does that. And in the ensuing, I don't know what this has been, 7 years, 8 years since then, the GHK has been through all these different changes, right? So that version 1 is gone, and then there's version 2, and version 3 is retired, and then version 2, and then there's version 3. And then, so then that was all GHK1, and then now there's GHK2, which has a completely new, way of doing things. And then, no, that one's gone. Now there's GHK3, and if you're up to date, then there's GHK4. And so, over this time period, this has changed 5 or 6 times. And these are not just sort of hey, I install a new version. It's there's new command line options. There's totally new ways of doing things, totally new pipelines. And so, if you had built this by yourself, and you would have had to update every single time. And so what frustrates me is, as a community, we are all doing this ourselves on all our pipelines everywhere, building and updating and testing and validating everything. Come on in, don't be shy. There's grab seats. So the goal of bcbio is to try to avoid everyone doing that themselves and working it together. And so the other sort of matrix is there's things are changing, and then people want and expect and should expect a whole bunch of features. And so this is another figure that is meant to be there's a lot of things and you're not supposed to read them. This is from the Nextflow paper, where they compare Nextflow to other pipelining systems and sort of describe what it does and what it doesn't do. There's stuff from where it runs, there's containers, what schedulers does it support, and the point is not any specific thing, it's that there's a lot of things people would want to support, right? So you write your pipeline, it runs on, your local cluster, which uses SG, and someone you want to give it to someone else, the first thing is they're not going to be using SG, of course, and using something else, Slurm or something else. So you're like, does it work on that? Then you want it to work on Amazon, does it work there? And you can imagine sort of the spiraling set of requirements that evolve over time, and which is why, you know, tools end up not being very difficult to sustain, because you sort of build up this set of features that you need, and then it's too much and it collapses in on itself. And so to demonstrate that we have not yet collapsed in on ourselves and we are still here, this is sort of a, you know, some examples of how we're continuing going over time. So here's bcbio. This is a screenshot from April, I guess, but you know, back here in whatever, 2010 or something, we started working on bcbio, and you know, this is a commit graph. So when it's green and up, it means things have been happening, right? And so like over those time, we've been pretty steadily keeping things going and adding and improving things. And the other aspect is sort of helping people make things work. And so there's, you know, over this 1,400 closed issues, and you know, we help people when they have problems, and try to do our best to do that. And so, you know, it's a fully active open source project that we want more people involved in, which is why we're here. And so the thing that has changed a lot, if I talked about bcbio last year versus this year, is how we represent the workflows and the things we're trying to do to make it work even better with multiple tools. And so we have pretty, like everyone else, like that big matrix, like pretty hefty infrastructure goals, right? Like you'd like the pipeline to run on local machines, you can run something small. You'd like it to run on a cluster. So, and then there's a million different cluster schedulers, right? You'd like it to then, so our cluster's busy, you'd like it to run on a cloud. And then, you know, I have a lot of other goals beyond this that I want to happen that are hard to make happen. So I would like to run in clinical environments where you have a locked down set of requirements. I'd like to have a nice Galaxy user interface to it, so that, you know, researchers can run the analyses without having to know any command line stuff, and then edit configuration files and other things like that. I'd like it if LIMS could, you know, easily enter the information. It just fits into a bcbio run that runs automatically. And I mean, I'd like it if a regular person who got their genome sequenced at Veritas or somewhere else where you can get your own genome could take it and run their analyses themselves. And I'll talk about that with Personal Genome Project later, sort of how trying to make that happen. But these are way too many things to ever happen, right? So I'm by myself or by ourselves working on the project. This is a lot, right? Like this is a whole lifetime of work for a whole set of people to get things going. And the reason that want to make this all happen is that increasingly you spend a lot of time moving stuff around, right? Like just advanced bound facts. There's a Nick Lohman quote about bound facts being advanced data copying. And people have been talking about forever that we shouldn't do this, right? Stop copying data around. It makes no sense, right? Take your analysis to where you want it to go. And so to demonstrate, I got motivated by this from last year because I saw Aaron Quinlan, who's an awesome guy, wrote his lab does bed tools and a bunch of other useful software. You know, it was being like, you know, the only way we could scale is if we stop doing the data. And so I was curious how long people have been talking about this, and you can find these quotes, you search on about moving data from, you know, you can see like quotes from DNA Nexus from 2013 and people from 2014. We got to stop moving the data around, come on, you know. And, you know, in reality, we still do this all the time. I mean, you take your data and you move it to where your computer is and then you run it, right? And the reason is that it's a lot of work to set up and configure analysis. So once you have your system running, right, once you have your local Galaxy server, once you have your pipeline working on your local HPC, and your data happens to live somewhere else where you don't have that environment set up, you don't want to move the analysis pipeline there, right? You say, I got it set up, I got it done, it's good to go here. And, you know, they'll have some other different analysis pipeline they prefer running that you don't know how to run. So you end up just porting it over. And it's because it's work, right? To take something that scales and then put it into a totally new environment. And the reason we end up with all this different work is that there's a lot of different great workflow systems. So, if you're going to other training later today, you could hear about all these. I think Nextflow, Pablo's here. I mean, and this is an awesome workflow system. There's Galaxy, right? Of course, which is another awesome workflow system that you have a whole user interface. You have an entire huge community ecosystem built up around it. You have SnakeMake, which I think someone is talking about later on, which is another awesome workflow system that has its own way of defining dependencies and way of running things, and people very much like that. So the problem is there's a lot of these workflow systems, right? And they're really, really good. And so the common workflow language helpfully has a incomplete list of existing workflow systems. And I don't know, whenever I looked at this, there was 181 different sort of workflow systems there, which is a reflection of that we like to build workflow systems for some reason, and a lot of them are built and not used or used for only a little bit. But I think what it's a reflection of is that we're as a community, as a set of people, it's instead of fighting and being like, stop building so many workflow systems, I think you can say, we're never going to agree on one workflow system, right? Like, there's advantages and disadvantages to each. Some people prefer a text-based, some people prefer a user interface, some people need some specific features one has, and then some people are just familiar with it, right? Someone taught it to them when they were in school and they liked it, and so they kept using it, and I don't want to learn a new one. I think we should just accept that this is the fact. But what it does is creates a huge barrier to working together, right? So if you're working with a group and they have a Galaxy server stood up and you use Nextflow, then you can't take the workflows that they wrote on Galaxy and use them on your Nextflow system, right? And vice versa, you can't easily take your Nextflow system and give them to them and they can run them on their, so you have a barrier, even though you're all using open source community built with a big, community around each of them, you can't share with each other because you can't interoperate. And you can't take something that they wrote, say like, oh, hey, they have a great variant calling pipeline, I'm gonna take that part of it and then mix and match it with what I have and put them together and make something awesome. I don't know. So these all, I would like to do better, right? I'd like to do, and we are no better in bcbio. So bcbio is moving away from its own sort of internal workflow system built on Python. And it was the exact same problem, is that, if you have something in bcbio, you can't easily take that and run it on Galaxy. You can't easily take that and run it on Nextflow. And it wasn't the intention. I don't think it's anyone's intention to do that, but you just end up in this state. And so to do better, we need better abstractions, right? And so the outcome of the I don't know how many years ago it was, 4 years ago, 5 years ago in Boston, the Max Open Source Conference had the Collaboration Fest, and a bunch of groups, Galaxy and Seven Bridges and Arvados, and a few other people got together and they said, why can't we share our workflows? Like, what's our problem? And our problem was we don't have any way of defining those workflows in a standard way. Everyone wants to interoperate, but there's no way to do it. So everyone incidentally builds their own, and it's a pain to map between them and stuff. And if you're staying for the Collaboration Fest, there's also, I don't know if any people from here, but there's the NIH Data Commons is going to be here as well, which is a big consortium of groups that have all built their own workflow systems and their own stacks of things and they sort of interoperate with each other, but not really, and they do very similar things. And so it's a common problem, right? Like we all want to interoperate, but we're not, we didn't have a way to do it. And so the way to do it is Common Workflow Language, or there's also WDL as well, which are two sets of standards which are doing the same thing, which is defining explicitly how you run a workflow, and then and then put it together. And then that's one step. And then there's the other side is your data and your tools. And so there's Docker and Singularity and other container magic that's helping things work together. So the point is we're having these better abstractions and things are getting better. And so, Common Workflow Language, if you're really interested in this, you should go to Michael's tutorial, which I think is right after lunch. But essentially, it's a way of defining workflows. So, there's a workflow, there's sub-workflows, there's steps with tools inside of those workflows, and you're essentially just defining how to run a set of things in a standard way. And it's built specifically to be portable to multiple systems. And I'll show in some practical examples how this is useful, why it's awesome. And I mentioned WDL as well, which is these are the two sort of communities that are coming together, hopefully, in a way that will make these definitions interoperate 100%. And WDL's the exact same thing. It's workflows, sub-workflows. How do you define the tasks? CWL has more of a focus on sort of portability. WDL has a focus on being writable by users, and so hopefully WDL will kind of be like a front end to CWL as they group together. And WDL is now Open WDL, which is an open community, and all I do for that is just be like, we should work together with CWL, we should work together with CWL. But that's a great community, and they have I'll talk about Cromwell from the Broad. They're one of the drivers of it, and they have that runs both WDL and CWL, so they have them all interoperating and working together. And okay, so I feel like I'm talking a lot, right? So is it are we good? Is this helpful so far? Yes. You have questions or keep going? Yeah, okay, keep going. Yell at me if I. So that's the motivation, right? Is for everything. I don't know. How can we work better together, and what can we do practically to do that? And so what I'm going to show here is how workflow abstractions are awesome and really help you do really practical things using sort of bcbio as examples of how that can happen. So this is all stuff that bcbio can do right now, which I think is very hard to do with a lot of systems, right? So it can integrate with multiple platforms. So you can run it locally with, there's a few runners. I'll highlight one. Toil is a nice one from UCSC that runs locally. It can run on HPC or locally or in cloud with the Cromwell runner from the Broad. And then it can run on all these different platforms, which are more like sort of hosted platforms that handle data management, data provenance, tracking, graphical user interfaces, all this stuff, Arvados, DNA Nexus, and then Seven Bridges, which is also Cancer Genomics Cloud. They're the same platform. But what this is doing is now bcbio, now that uses CWL, won't have specific infrastructure, right? So there's no longer a runner for bcbio. bcbio isn't a thing that runs things. It's a thing that generates really good workflows that are validated, that run well, that then you take to your platform that you prefer and run it there. And this lets like the community of bcbio, which we're developing or is developed and is continuing to develop, focus on biological problems. So right, instead of the questions being about how do you run this or how do you do that, or can you fix this on the runner, then the questions are now about biology, right? This doesn't work as well. I'm calling RNA-seq. Here's a validation. It doesn't work as well as it should. How can we make it work better, right? And then you fix that, and it can then run on all these platforms everywhere. And so, because we're at the Galaxy and BOSS conference, I want Galaxy to be on this list. So that's hopefully one of my continuing goals, to bother everyone to make this happen, so that you can add Galaxy here, and we can have that working as well. Because I've been wanting to run a bcbio on Galaxy ever since I was not able to do it for a long time. So hopefully that will happen at the end of this conference. It won't be all set. And I want to show you a really practical example of why CWL is useful. That's happened in the sort of lifetime of bcbio supporting CWL, which is a yearish time frame, right? So I used to use a lot this rabbit executor from 7 Bridges, which is awesome. It's an open source tool. And about a month ago, or not a month, I don't know, a week ago, 2 weeks ago, something like that, they said, we're not going to support the open source version of Rabbix anymore. We don't feel like it does a lot more than CWL tool does. We'd rather work on that and work with them on that than have 2 separate things, right? So you can imagine how crushing this is if you are 100% reliant on this as your runner, right? Like this is like when I built my entire set of things around GATK, and then they were like, oh, we're not going to be open source anymore. Sorry about that. You've built everything on top of it. Then you have to rip everything out and start in some new way. And so this is why ever since I've had bcbio be a Python thing, I've been afraid to switch to something else like Nextflow or SnakeMake or all these things that exist, because if people make decisions, then you are up a creek, right? But the advantage is that then within that is a month ago, right? The 25th of May. So just over a month ago, right? So the Cromwell team announced they're supporting CWL with Cromwell. So yay! And I'd gotten this working before in that sort of 2-week interval between there. And so I'm like, oh, Cromwell is working great. This is really nice. It's a nice runner. This actually is a lot of advantages over Bunny. And so, but I didn't have to change my workflows at all. Like I didn't change what bcbio does. I didn't change the biology. I didn't change anything. And I've essentially upgraded my runner to something that's supported by the Broad, which is a big place and hopefully not going out of business anytime soon. And it does a better job, and it's being used in a lot of places. And this is, I think, an amazing thing about CWL that you don't have with any other things, is that you could go through this and be completely fine and actually better off, which is cool. So this is practical on the road CWL. And so what I'll try to talk about what it can do, and that I think are the goals right now, which are taking the exact same workflow and running it in multiple concurrent production environments. So I think you should be able to have a workflow with bcbio that you're running on your local HPC regularly all the time, and you're happy with that. And then I think you should be able to say, we don't have enough compute, we have a big project coming in, the HPC is gonna have a 3-week downtime because of whatever, right? Like, we should be able to then go, we need to run things, we should be able to take those exact same workflows we've been running, the exact same versions, and run them on DNA Nexus or 7 Bridges or Avatos, and pick up that slack while we need extra stuff to do. And I think you should also be able to say, okay, I don't want to use a provider, I want to just I want to do everything myself on AWS or Google or Azure. And so these are all things that CWL makes possible right now. And what it gives you is this coordinated release and update process. So you've got 3 things, right? You've got a workflow that you define, and this in common workflow language. You've got your tools inside some kind of container or some way of organizing them, and then you have your data. And you can then, if you keep those things the same, right, you can then put your things back and forth between all those places without missing a beat. And so I think that's a unique thing about CWL, lets you do that and lets you have choices about what your runners are, right? So I can be agnostic about whether you prefer DNA Nexus or Avatos. Just you can give it to them and you can do that same thing in multiple places. And so, wow, how did that? There's like that's cool. We'll see the whole thing there. It just disappeared. But anyways, this doesn't mean anything other than like what it's trying to do is bring together these connections. So I think of each of the groups. There's supposed to be another box up here, which would be 3 of them, which would be more impressive than 2, right? But 2 is okay. But I think there's like these awesome communities where we're doing a great job and things work together. What we need to do is bridge these communities, and so what these common workflow language is allowing to do is bridge those connections and make things work together. So that's all my motivation, right? Are you guys fired up? Like you're ready to punch through a wall and like I don't know. That's motivating everything. And so what I thought I'd do is then talk about some practical things, sort of that bcbio does that this CWL is enabling. So show how it actually works and under the covers. And what time do you guys want to take a break? Like 10? Yeah. 10:15. Okay. I don't know how else to go for. Cool. Just let me know if you're bored. So.
You drinking water either.
Yeah, I should drink some water. That's a good idea. Don't get dehydrated. So, so CWL in bcbio, this is how it works. So you start with a high-level configuration file. If you've used bcbio for not running with CWL, it's exactly the same as it was before. So you define the way your workflow in the exact same way. We try to be really careful about sort of whatever you learned before, you can then take to CWL. It uses that to then generate common workflow language. So you don't have to write common workflow language. If you go to common workflow language tutorial, I'm sure that Michael will walk through like what common workflow language looks like. You can see it all. Within bcbio, it's an implementation detail essentially, right? Like it takes your configuration. You don't have to know about it. It builds the CWL, and then you will send it places. So it's good to know that it exists so you understand what you're sending places and what's happening, but you don't have to write it or do anything else. And then the idea is that then any infrastructure that supports CWL, you can take that workflow and put it there. So the three things you need are the generated CWL, then some sort of way of getting all your tools and the bcbio itself, which is either Docker or Singularity, or you could have a local installation of bcbio like normal, and then your data. And so there's a tool called bcbio VM, which is, so all these things are very cleverly named, right? So it's sort of a CWL wrapper around bcbio. So bcbio is doing the running, and then bcbio VM is doing the sort of management and building things up. So it essentially, it's a tool that you install from Conda. So it's bioconda. I think there's also a bioconda tutorial later today, which is an awesome community of people and there's thousands of tools on there that all work together, and bcbio is one of them. But it has a lot of dependencies, but you don't got to sweat that because you just write one command and they all just happen. Conda is awesome, and I'll use this to try to show. So that's sort of everything. And then what I'm going to try to do is show an example of how you could write a workflow and run it. And so for the example, I'll use some data from the Personal Genome Project, which, if you don't know about this, is an awesome project from it started at Harvard and then with George Church, and then it's kind of a big community now in multiple places. But the idea is that people can get their genome sequenced and then share it with the world along with their traits and anything else they want to share. And then you can take that data. It's completely unencumbered by by restrictions, data use restrictions, and so you can use it for validations, you can use it for testing, you can use it for analyses, you can take it and make a software product out of it and make a bunch of money, whatever you can figure out, you can do it with that. So it's kind of a really cool idea. If you've ever had to deal with like trying to get someone else's data, do analysis, you know how nice it is to be able to just have a whole set of genomes. And if you live in Boston, a lot of these PGP. Oh, go ahead. Sorry.
I was just going to plug Open Humans, which is a spinoff.
I have it next to it. 2 slides over. Yes, but Open Humans. Yes. Yes. So there's also this is tied in with Open Humans as well, which is a good plug. Which is Madeline, Madeline talked to us. She had one of the keynotes at BOSS last year, and Bastian is also, I think, with them as well.
Yeah, he's going to give a poster.
Yep.
And a lightning talk.
And a lightning talk. So there's former PGP people that are now Open Humans, which is trying to make this data, enabling this data to be used. And so it's I think it's been really good. Like the Genome in a Bottle initiative, which does validations, has taken PGP samples and used them, and it overcame a lot of like consenting restrictions on some of the 1,000 genome samples. Like what are they exactly consented for? because everything is freely consented. And yeah, if you live in Boston and there's a lot of people happen to be from Boston because of sort of the initial recruiting phase. So you can sometimes meet people that are in this that you only know by their numbers. And so for this example, I will talk about James Vick, who has identified himself, but he's one of the earlier PGP participants, and HUG7 BBF if you like the IDs and everything. But unfortunately, most of the PGP people white male, so there's not they need a lot of work on diversity in terms of that. But he's nice to work on, for an example, because he has a ton of different information that he shared about himself. So here's Open Humans, right here. He has a profile on Open Humans as well, and he has all kinds of data that he shared, both the surveys from the PGP as well as there's all kinds of like bacterial kits and fungal kits and other things that you can microbiome type things. So really cool sort of data set that has both health information and also this other information that you can then try to put together interesting things about. So yeah, it's a really great community of people. And so this is ID, and this is where we get into sort of talking about how to run bcbio. So you had asked before what a bcbio configuration file looks like, and so here it is in all its glory. Right here is how you define analysis. So bcbio tries to be very high level, right? So essentially it's a description of what you want to do. And so you have your input files. This is a BAM file from that spin all the sequencing available data is Veritas recently resequenced a bunch of them, and it's all publicly available. And you describe what you're doing, you type analysis, you want to do variant calling, what genome build, I want to work on build 38, and then you define what sort of things you want to do. So I want to align with BWA, I want to do variant calling with GHK haplotype caller, and then I want to run 3 different structural variant callers, Manta, lumping, and CNV kit, and I want to call HLAs without any type. So I'll walk through what this means if you're not a variant calling person in a sec, and what the tools are. But this is the high level description and of what happens. So there's no you can get in and tweak if you want to tweak. If you really like tweaking parameters, there are options to tweak things. But in a general case, you should just be able to say, I want to run these tools, and it will run them in a good way for your input data. And the other component is then finding the data where it exists, right? So this is the CWL portability, like where the rubber hits the road, is figuring out the data and where it lives. So if you have a local file, there's another YAML file, which is very similar, where you define where the data is. So here you're defining your local data, the folder where you have the reference data, like HT38, the reference and all that, and then folders where you have your inputs. So these are just, it could be anywhere, right? But it'll search within those, find the data, and import it. And then you define resources, so how you want to run, what type of system you're running it on, right? So 8 cores and 3.5 gigs of memory per core, things like that. So you're defining what's happening.
Brad.
Yep.
That configuration file you showed at the beginning, if you do want to tweak that a bit, are you actually changing that configuration file, or are you just changing the tools that it's running to go a different way?
Yeah, so you can just change the configuration file.
So you put options in there for certain things?
Exactly, yeah. So if you don't like GHK4 and you want to run something else, you could change it for Strelka to.
Well, I'm talking more about using different options within those programs.
Yeah, so there's also a way to tweak options. So there's a resources flag, which then you can sort of pass specific resources to specific tools. And so, right, if you wanted to pass something specific to GHK, you could do that as well. Yep. So that doesn't take the idea is that you can there's also a whole configuration page of like different tweaks you might want to, like more general tweaks you might want to do. Like, I don't know, I'm doing low pass sequencing, or I'm doing other things that we then try to do the right thing for those cases. But yeah, you can also go all the way down to like, I want to change this parameter to PWA in this way. So here's how I do it. Cool, yeah. And then, so this was the local. And what I want to kind of demonstrate is that if you're going from local to remote system with CWL, it's a very similar type setup. So here's this, it's an equivalent sort of how you find your data file on Arvados, which is a remote platform. Like, it's going to have the data uploaded, it's going to be available there. And so the resources down here, it's the exact same thing as before. You just, you talk about cores, memory usage. But now when you have your reference and your inputs, you have, instead of local file system references, you have references to these buckets on Arvados where the data is stored. And so, the configuration, the other configuration doesn't change. What bcbio does is now it knows how to talk to Arvados. It knows those inputs. It looks up the data there. It fills in the CWL in the right way, and then you can submit it there. And so, in your side, it's really just pointing the data to where the data is, and then everything else gets taken care of. And I'll show you how taken care of works. Here's I mentioned bcbio VM is the tool. Here is how you would generate that. So, the bcbio VM, you're generating CWL, and then you specify that system config. So the one I just showed, this is an example around Arvados, bcbio system Arvados, and then this is the description of the analysis you want to run. And so that's it, and then it's generating common workflow language for you that's ready to run. And so what it's trying to do is hide all the common workflow language, or abstract away the common workflow language, the details. You're just telling it where it goes. And then you can similarly abstract away the run process. There's a bcbio VM again, CWL run, and you're telling it you want to run on Arvados, and there's a bunch of different choices there. And this is the workflow that you generated in the previous step, and then you're telling it, hey, this is the project I want to run it in on the Arvados system. And so then, you can look it up there. If you don't believe me that it ran, because it did, and it was all good, I'll show you. But it's again abstracting away all the, like, how does a job get submitted there? How does, and it does it all for you, right? So it'll just launch there, and then you're ready to go on that platform. And this is, I mentioned you might not believe me, so here is a screenshot of it working, right? So that you would really, really believe me, because I have a screenshot. But these platforms are real nice. If you're used to, like me, like running, everyone wants their analysis run on their HPC, and you don't have any tracking and all kinds of other stuff. It's very nice. You have timings of all the different steps. You have, like, you can drill into any of these and look at how they, all the logs and everything like that. It's quite nice. It's quite nice to having a managed platform. And the magic then is, if you don't have that managed platform, if you have it on locally, this is the command to run it locally on HPC. So bcbio VM, CWL, system config, now bcbio system local, and that same sample file. And so essentially all you're doing is subbing this system configuration of one for the system configuration of the other. And so what you've learned about how to run things is applicable to any system you want to take it to. bcbio is just kind of doing the thin wrapper around interacting with those systems, or your local file system, or whatever. And then the magic is all happening on the back end. And so similarly, if you want to run this, it's, again, almost the exact same thing that happened before. Now this is an example running with Toil, the Toil workflow engine. This is bcbio VM CWL run, same as before. Now you put Toil instead of Arvados, and you point at the workflow you've developed. And it then launches Toil, runs the analysis. You'll see it going. And to belabor the point, because I think it's cool, is this is if you want to then submit that to your cluster using Cromwell. And so it's bcbio VM, CWL run, Cromwell. the workflow engine, and then stuff you would have to pass to your scheduler. Like, so in this case, you're not using a container, you're just running a local bcbio installation. You know, the queue you want to submit to, it's a Swarm system. You want to set the time limits, other resources. But, you know, ignoring the, like, hey, this is the system itself stuff, essentially you're doing the same thing over and over again, right? You're doing bcbio, VM, CWL run, the runner you're using, and then the workflow itself. And so this is a practical, real workflow that then is running, I don't know what to show, three different tools, exact same thing, exact same setup, exact same configuration inputs, running in all those different places. And you can get, so I can continue to show off, I think, for me, you know, you can then run this on DNA Nexus using almost the exact same configuration. So here's a new system configuration, right? Here's your resources. Pretty much same thing, you know, you can tweak it how you want. But then, you know, in DNexus, you know, what project are you running in? You know, where are the references? They're in. And DNexus, there's like a shared bucket, so it's a shared project called bcbio resources, where the reference points are already loaded for you, and then your inputs are in, say, data input. And then you're doing the exact same thing. You're gonna then run it on DNexus. And so DNexus is to show that everything is not exactly the same. Is fancier, I guess, in a way, in that it has a applet that we built that will actually run everything there. So you don't have to even have bcbio installed on your system. You can just upload it there. And so this is showing, you know, if you know DNexus at all, there's like they have this command line called dx, which is that sort of command line interface. And you're essentially taking the input file, the project and the system file, you upload them there, and then you use this tool, gives this dx again, dx run, and there's an applet right here, applet's bcbio run workflow, that you tell it, hey, these are all the different files I wanted, and it just runs. And so there's a wrapper that sort of does everything for you, so you don't even have to install bcbio VM on your own system. So you've now taken this inputs, right? So the exact same workflow, and run it on DNexus, on Arvados, on locally with Toil, and on a cluster with Cromwell, right? So I think that's pretty cool. I don't know how do people have questions about this? I feel like maybe I'm going through a lot very quick.
Take a break.
Yeah, sure. Yeah, sure. That sounds good. So yeah, let's see, where am I at for taking a break? Yeah. Like I'll look through this, and then we'll take a break. That sounds good to everyone. I can use a break from talking. Cool. But DNX is also, if you're still like me, who you don't get a lot of use on like platforms like this, it's pretty cool. You get like these graphs of like, hey, here's the whole workflow running from beginning to end, and you know, you can tell where things take a long time. Right, here's alignment, here's variant calling. So you know, you can see sort of all the different steps, what's happening, how long they're taking, how it's walking through there. You can dig in and see within within runs, like where things specifically are happening. You know, how the different steps are broken down. You can see things like this, where this is parallelization of the variant calling over multiple regions. You can see cases where, hey, you know, it's working real good, like it gets broken into, I don't know, 1, 2, 3, whatever, 8 steps, 7 or 8 steps. They all run pretty similarly. You can identify when things don't work good, right? So here's a case where you get broken into 7 steps, and most of them take 20 minutes, and then this one takes 4 hours and 40 minutes. So you can see, so when you have something that takes a long time to run, you can easily see, hey, you know, something's off with this region. I wish it didn't take so long to run that. I wish it wasn't waiting and waiting and waiting for that to run. You know, this is an example of fixing it, right? So you're sort of fixing it is it's now an hour instead of 4 hours, so a little bit better. Using the magic of GHK4 Spark parallelization, you can fix things, but this is just kind of cool to show, like, you can take that same workflow, you can run it places, you can find things on one, fix things, and then they will be portable back to any of the other workflows that you're running. So it's nice to have, you have the advantage of having all these systems is then you have a lot of different knobs you can turn and learn about. Cool, yeah, so that's probably actually, we'll take some questions and have a break, because then I thought that's sort of how it ran and like the nuts and bolts of making it happen. And then for the next part, I thought I'd walk through what the outcomes look like, what the variant called stuff looks like, show a couple of useful examples of how you dig into a variant or look at specific interesting things within those outputs. Cool.
Brad, I have a
Yeah.
question.
Yes, thank you.
I see a bcbio includes a lot of tools.
Yes.
Say I have my own bioinformatics tool I want to run with the bcbio pipeline. Is that possible, or is it easy?
Sure. I mean, definitely. Yeah. I mean, what bcbio is trying to do is give you the tools to make it easy, but yeah, it's just inserting them into your path. So if you have your own specific thing that's not in Conda that you want to include, that's, as long as you can call it, you can run it. Yep. And then bcbio just calls out to it. So there's really no magic in terms of bcbio includes it or doesn't include it. It just makes it easy. And then if it's a useful tool, then the idea is you roll it in, make it, have bcbio install it, and then it can be available to other people as well.
So are you saying you will need to install the tool to bcbio? I'm not sure I get.
Oh, sure. So I'm just saying the tool has to be installed somehow.
Included by bcbio.
It doesn't have to be, like, if you wanted to run your own thing and test it out and whatever, then your tool isn't wrapped and ready in Conda, then you can just install it however you got it installed and run it, no problem. If it's a useful tool, then generally useful, like the idea of bcbio, like, is to build this, is to make it available so other people can use it. And so in that case, yeah, the goal would be try to wrap it up in a bioconda package, install it, and then have the integration be part of bcbio so that other people can use it as well.
Non-Python tools?
Anything, yeah, I mean, anything from bcbio is written in Python, but it's just calling out to command line tools. So yeah, I mean, there's, yeah.
But can you wrap non-Python stuff in bioconda?
Yes.
You said you have to call it conda.
Yeah, Conda can install anything at all. Yeah. So like, I mean, there's tools that use like .NET, that things that, I mean, a lot of Java things, and a lot of things that use pretty much everything. Yeah. Yeah, Conda, like, although it's like originally a Python installation system, it's really just like it's a glorified set of shell scripts that install precompiled binaries in the right place and figure things out. So as long as you can get it to compile within Conda, you can install any tool. Yeah. It does. It just, the magic it does is getting all the dependencies right for you, which is pretty
And
good.
it also runs in Windows and XPSD, right?
Yep. Yeah, across a lot of different platforms. Yeah.
Same way.
Yeah, so Conda is awesome. If a big plug for Conda, if you're interested, there's the Conda tutorial later, I think, which is worth checking out. Cool. Other questions about this part? Cool. Yeah. So let's take, I don't know, take 15 minutes and get some coffee and water or whatever, and then we can come back and and we'll do some personal genome project action and. Sound good? Cool. Thanks, guys. Let me just pause this.
That's
Get ourselves back set up.
Cool. So everything's going good. You guys, I was happy everyone talking to each other. Thank you for the.
What is that, dude? Hold on. I don't want it to auto advance on me. That's not gonna work for me. Go
go as fast as possible. So yeah, people, thanks for the questions. If you have questions, feel free to speak up during this for sure. I think one general thing people ask about CWL, if you do CWL, the stuff that I showed where bcbio is wrapping things, it's just convenience, right? So bcbio is like doing the stuff to call the tool that you don't have to do it. But if you're an expert in running your own tool, the bcbio CWL is just like something you take and run. So you don't need the wrapper. You don't need to, if you're an expert in Cromwell, you can just take the CWL and run it. You don't need to use the bcbio wrapper. So if you're not, then that's what it's there for. So it's kind of meant to, that's what's letting it be no longer a platform, right? It's like, some of these things are wrapping, but it's not a platform itself. It's just helping you out. And if you have your platform and you're ready to go, like, we're going to be able to give bcbio to you. Cool. So I thought like that's sort of like all the sort of like techy stuff about how things are working. And then I wanted to do some sciencey stuff about doing analysis and writing things. And so this kind of walks through a variant calling analysis, and hopefully it's useful. I kind of go from the beginning where it's like, hey, here's what variant calling is, to showing the examples. So if you already know, just bear with me, just in case people don't know, just so I get everyone on the same page. And then, so I'll overview that. I'll show an example of just an n=1 analysis with the PGP data I mentioned earlier, and shows interesting looking at the small variants for Alzheimer's risk, HLA calls, how to get those, and then some structural variants and sort of how you visualize and look at structural variants. And so just to kind of set the tone, I hear there are other organisms out there, but the only one I get to work on is human. And so this will be a human example. It's kind of an inside joke because my degree is in plant biology, and I always wanted to go and work on plants again. But the only crazy stuff I ever do in my day job is like maybe I'll get called from variants on mouse or rat or something. But I never get to do cool genomes anymore. Just human. But so this is the human genome if you're not used to looking at it. And
well, we are using bcbio for the mammals and elephants.
That's true. So bcbio runs everywhere. I should be clear, right? So bcbio runs on a lot of different organisms, and other people in my group get to use it on like cool things like mammoths, and you get to run like the elephants and woolly mammoth and stuff. I never get to do any of that because I'm not cool. But yeah, you can install a dog genome. People do a lot of things, and so it runs on any type of organism you want. So that's a good point because I didn't mean to say that it doesn't run. I just don't get to do it myself. And so you got the genome. Typically, what we're doing now is doing Illumina sequencing. So you break up the genome into smallish chunks and you sequence them. You pile them up with alignment step, and then at each step you have all those aligned reads, and you essentially just walk along each position and see whether they're different from the reference, and you call it. So this is a heterozygote, AG heterozygote. So at the simplest stage, that's essentially what variant calling is. And you have a bunch of different types of variants that you can detect. So you have substitutions or single base pair SNPs. You can have deletions, insertions, inversions, and all these small events you can detect with a small variant caller like GTK. And then you get the cooler, bigger events, structural variations. I'll talk a lot about these because for me, I wish we spent more time thinking about these sorts of problems. They're very hard with small variants, small reads, but we are getting better technology, right? PacBio is getting cheaper. Oxford Nanopore is awesome. There's a lot of cool things coming that we will be able to look more at structure variants, and I think we will learn a lot of important things about human biology and every biology, I don't think for humans, because the cell doesn't care if it's a SNP or a structure variant. It can make each of those, it's all good with making either of those. but there's a lot we miss because we're not actually checking these. So that's why I would talk about the structure variance stuff. But there's all kinds of crazy stuff that can happen once you start looking at structure variance. This is a nice overview of some of the craziness. This is the simple stuff here. You know, you get those big deletions of chunks. You can get insertions of random things. You can get big chunks inverted. You can have tandem duplications or duplications that are spread out, or and you know, tandem duplications that are copy number variants. You can detect. So there's lots of cool things that are happening. GATK is the example I'll use here for small variant calling. So it's I think there's a tutorial about that. I don't know if it's happening now or happening this afternoon. What's that? Afternoon. So if you're interested, they do an awesome job of explaining GATK and how it works and the awesomeness. It's a really nice tool from the Broad. And I showed this before, and here it is again. This is best practices. You know, they not only, this is I think one of the unique things that Broad did, which was awesome, is not only did they give you a tool, but they told you how they recommend that you use it. And you know, if this is a lot more tools do this now, but this was sort of one of the first ones to do it, which is why it's so popular. Because a lot of people would give you a caller and be like, "Good luck," you know, and then it would spit out a lot of variants, and they would say you should filter them to something that makes sense for you, and they wouldn't give you a recommendation on how to do that. This is the best practice is awesome. It was an awesome improvement in the field, and so the magic that it uses underneath is this thing called haplotype caller, which I think you should go to their presentation later rather than me explain all about it. But essentially what it does is looks through, I talked before about, hey, you just pile things up. And that's true, you know, sort of, you pile things up. But what happens is you get these piled up things that are close to each other, and how they're piled up and then aligned that affects each other. So what GATK does is it piles up the regions, figures out where they're at, and then it says, okay, here's some active regions, it calls them. And then within that active region, instead of just looking at each position, I can actually look at that whole thing together. It, you know, assembles into a graph and looks at possible options, which ones make the most sense. You know, ones that are probably sequencing errors like this get pruned up, and so you remove some errors that way. And then, you know, it does magic to determine the likelihoods of each of those, and then actually does the calling. So just to say that, like, you know, what's happening now is a little more complicated than just piling things up and then looking at them. So don't write your own variant caller yourself. And, you know, and then once you've called variants, you want to try to place them within the genomic context. And so there's a lot of tools that do that. The two major ones are VEP, which is the variant effect predictor from the Ensemble team, and SNPF. Both of them do very similar things. Is it looks at, you know, it has the set of transcripts in the genome, and then it looks at your variant within the context of those, and then, you know, says whether it's you know, hey, it's causing a silent mutation or a non-silent mutation, and then ranks them in terms of how likely it is to have an impact on, say, a protein coding region. And so this makes it nice if you're, you know, sequencing a genome and you have someone with a disease, and the first sort of pass you can use is to say, hey, what are all the things with high impact, and then filter based on those. And so to do those sorts of things, you know, sorts of analyses, there's nice tools that are built on top of that. Gemini is one from Aaron Quinlan, who I mentioned earlier, his lab. And the nice thing that it does is it not only takes those effect predictions, but it puts them in the context of all kinds of other external information. So this is human stuff, but like there's a million different, like, you know, associated databases where you can get frequencies, and then there's a bunch of sort of standardish queries that you might want to do on top of those. And so it makes those easy to do. You can do sort of any custom query you want, or there's queries for, like, within a family. So you can say, yeah, I have, you know, affected, unaffected parents and siblings, and I want to pull out, you know, variants that are only in the affected. You know, you can look at tumor normal cases. You can look at sort of control, bigger cohort studies as well. Nice tools that make it easy to do the downstream analysis. And what you're getting essentially, so if you haven't, I'm going to walk through some of these. So I just wanted to sort of describe what it looks like. If you haven't looked at BCF format before. It's the way that you describe variance. And I think of it like if you ever, I don't know if you ever go to the horse track and go to look at the horse racing forum, the first time you go, there's all these numbers and it's like, what is going on? And then once you learn what's happening, it's not that big a deal because you just know where to look and you know, understand what things look like. But you know, it's so there's some nice tools to help you if you haven't looked at VCF to help you weed your way through it. But essentially it's a tab delimited format because, you know, why not, right? And you look at a chromosome, a position, and then it has the actual change itself. So there's a bunch of complicated ones, but here's a simple one. You know, it's a A to a G. It'll have a quality score normally. It'll have these filtered, whether it passes or is filtered, and then other information about it helpfully in two different places, right? There's one here in the info field where there are key value pairs that are about the whole variant itself, and then over here are specific samples, so it's a multi-sample format, and you can then have key value pairs, except the keys are here and the values are here, you know, but the important ones are stuff like genotype, or quality and depth, other things like that. But so it's a line living way of describing variance. And so I have a couple examples so that, you know, and if you're really into like learning about these, which you should be if you want to do variance, this is a nice like VCF tools is like this poster that sort of describes how you would represent the different types of things. So you know, I mentioned the SNPs are pretty easy, C to T, but then like once you start getting into weirder things like there's insertions or deletions or, you know, these complex events where a lot of things happen, then it starts getting more hairy, and so there's a lot of some ambiguity in how it's represented, and then it also represents things like large structural variations and things as well. So you have like a position, and then it'll tell you where the end is. So this is a big deletion from 100 to 300. So I will have an example of a deletion later too. And so these are, if you like my description of VCFs and you want to learn more, these are the places to go to look. Broad has a nice like sort of step-by-step guide, which I think is really useful to highlight the useful things. And if you're really a masochist, you can look at the whole specification for how you represent everything and learn anything you'd want to be. And so what I thought I'd show an example of is ApoE status for the PGP patient that we talked about. So ApoE is it impacts a couple things. Alzheimer's is the risk of Alzheimer's is the biggest one, and it's also involved in cholesterol metabolism. And it's kind of a nice example because there's two variants that you can query that essentially tell you, and there's a allelic series of you know, how the two variants here, and then they give you different ApoE different ApoE alleles, and then there's a series of like how those are affected, and going from sort of a common one to you can have 11 times increased Alzheimer's risk. And so, you know, if you what bcbio spits out is a file, a BCF file. There's a patient, GHD haplotype caller, BCF.gz. So named with the sample and the caller. So you could have in bcbio you can have multiple callers. So one of the nice things with bcbio is you could call with 4 different callers just as easy as you can call with one. And so if you're interested in really seeing if every caller called it, that's why it's named with the caller. But you can query these positions. So this is a query on the first one, the RS429358, and it's at this position in HD38. And so what you get out is I had to split it up over multiple lines because so it fits. But you get this is the call right here, right? So you have on chromosome 19, that's position. It's a T to a C change. Then it's got the quality. And this is a pretty high quality. It's passing. And there's the annotations I mentioned before, so it's letting you know it's a missense variant. It's in ApoE, some other information about it, and then you can see the genotype is that it's 1-1, so that means it's homozygous alternate, and you can see the depth and other things as well. So it's pretty reasonable depth. And so that's that's a simple like TabX query. If you're using TabX, it's a nice way to just like, BCF is really good for pulling out a position. TabX is a really nice tool for that. It's indexed for that. Not so good if you want to pull out a whole sample at the same time, but one position is good. And then you can do a TabX query on the second one, and you get nothing, which means it's reference.
Very, very helpful, right? Because there's only the variance. And so if you kind of walk back to what that means. So the first one is the first position, and it's so it's a T to C change. So you have two C's there. So that's we can walk back over here. So that's this right here. So you got a CC. So that's the it's E4, E4, and then the other one. Sorry, is reference. This reference the second one, which is what CC as well. So you get 2 4s. So this makes me feel bad because I didn't realize before I did it. I just was using it as an example, and I thought there'd be nothing interesting. But this actually has a 11 times increased Alzheimer's risk. So he's got very bad ApoE alleles. So it's interesting the fact that you're finding something out, but it makes me feel bad because he probably already knows this. But so that's kind of how you would dig into a SNP variant file, right, and find something potentially interesting. And so if that's kind of, you haven't dug into these before, I have you could go to this link here, and it walks through sort of how you do the queries, what the tools are, all the sorts of things, just to give you a sense of like what you do to sort of dig into a VCF. And so, but I wanted to kind of get beyond that and show some of the stuff I think that's unique that bcbio does. A lot of tools call small variants and do a good job of it, and then you can query them. Some of the cool things that we have, I think one that I think is really useful is HLAs. This is another part of the genome that's super important and is often ignored or not analyzed because it's difficult, right? So HLAs are like the central component of your immune system, and so they're helping determine your immune response to a whole host of things. It's very important for things like cancer responses, other things like that. And the tricky thing about them is that there's a whole bunch of different copies. They're all repeats of each other, and so they're very hard to resolve with standard. You do a standard sequencing run and you try to resolve them, you're not going to get anything useful at all. And there's also thousands and thousands and thousands of different versions. And there are databases of where they have all these, and so you need like all those different representations to make good sense of it. And so the magic of the new build 38, which we use for this, is that it helps you resolve that because it can handle these cases. Previously, if you're using human build 19, you should stop and move to 38. But in 19, there was no way to specify if things are the same thing but at the same position. They would get poor alignment scores because they're at the same position. And what 38 tries to do is move towards this graph-based model of the human genome where we start to recognize that there's unique positions which are the same, and we need to know that the reads are aligning there in a different way because they're the same, not because of some sort of artifact or something else. And so because of that magic that's in BWA, you can then all these HLA, or a large portion of these HLAs, I wouldn't say all of them because there's a ton of them, are in one of the reference builds. And so when you align, you can then pull them out and then use a tool to do the typing of them. And essentially the way this works is to use 1,000 genomes build of 38, and so it has the reference genome plus all these HLAs, and BWMM will helpfully extract just the HLA reads that map to those and stick them in a file for you. You tell it to do that, and then you can then take those and then map them only to specific HLA sequences and call HLA types. And we use a tool called API type, which is one of sort of the more standard tools to do this.
it essentially it aligns to all the different HLAs. It then so it does a realignment to all the different HLAs. It then resolves what the most likely ones are because there's a ton of overlap between them. So, but yeah, you should definitely read about it. So it's a pretty useful tool. If you ever do HLAs, I think it's really useful with short reads, independent of bcbio. And so it's these. So the sort of at the end you get numbers, and so I don't know if the reveal is as exciting as the build up, but what I think is cool is that you get like this, you get 2-digit typing. So I don't think it's good enough if you like need like transplants or something like that, but it's pretty useful for just assessing what's there. And so I mean I think this is kind of cool. Like if like I'm sure that probably the James Vick, the PGP participant, has not had his HLA typed, or I didn't see it anywhere. So you can actually run this, and you can have his HLA types right straight away from the sequencing that he got. And you know, this is all part of sort of stuff that like bcbio is doing all the work here of extracting and running and then reporting. And there's a validation kit set that we use to make sure it actually makes sense. So it's a nice way to you enter one thing into your thing and you get it out for free. And so one thing a second thing I was trying to demonstrate here is like sort of the value of having whole genome sequencing over like, 23andMe typing or something like that, why PGP is useful. And this is one of those reasons like that, HLAs. So that's HLA. So those are two things we're getting, right? Small variants and HLAs. This is the other thing that I mentioned before that, because I think it's really important, is structural variants. And so I'll go into this a lot more than the other two, just because I think it's super important. But this is one of my motivating examples. Although this is a germline example, this is an example from cancer, but I think it sort of shows the point. This is pancreatic cancer, and they try to classify. This is a nice paper, just sort of about categorizing pancreatic cancer samples, and it classifies genomes from stable to unstable, right? And sort of with how many different sorts of changes you have. And so there's like, you can see sort of these different set of patients where you have these sort of local rearrangements and things like that, and then you start to have these like really scattered and unstable things over here that are affecting a ton of patients. And I think for me the eye-popping thing when I saw this figure was just how prevalent structural changes are once you look for them, and how much they impact the genome and when things that are happening, and how sort of blind we are sometimes to looking at that because we're looking at the impact of SNPs and other things as well. And I think the reason for this is that variant calling from small variants is hard. There's a lot of tools to do it, and it's hard. Then they all sort of have different sorts of sets of how good they are. But because it's hard, I normally use multiple callers. And so like for GHK, we use one because it does a pretty good job. But it's kind of nice to when you have a harder problem to kind of walk through have a bunch of different examples. And so we have 3 callers here. Manta, which is one from Illumina. It does split and paired end reads. So the way you detect these is the reads at the ends are the only evidence you have really is that the reads are doing something weird that is not normal, and I'll show you that in how it gets shown. And so they help do that. Though Manta and Lumpy both use that sort of evidence to call it, and then CNV kit uses read depth. So the other evidence you can have is if it's a deletion, you'll have less. If it's a duplication, you'll have more. And so you can run those 3 tools in bcbio. It all just happens magically under the covers with what I showed you before. There's no nothing hiding. That all comes out, and you get a BCF file. And so I thought I'd highlight just one call, which I think is interesting. Essentially, what I did is just look for things in the BCF in the call that, or in all 3 callers that they all call that had a predicted high impact. And so this is a chromosome 19 event, at this position, and it's a deletion. This is the Manta example, but
as I said, it's on all three. So it's you have a deletion. It's like almost 3.5 kb, 3.4 kb. Deletion, and then often the annotations will have some weird things in them. So it's kind of, you can take them with a grain of salt, but it's a deletion and then bi-directional gene fusion and things. But between this gene here, the KLK15 is where it's at, and then this is some of the transcripts nearby. It's probably just a deletion, not a fusion, but sometimes when you have two transcripts coming together, it likes to do it that way. And then it's it's genotyped as a heterozygote, and it's got pretty high quality scores and things, so looks pretty real. And so the next sort of step, when you're looking at these, is to try to then visualize it. And so I wanted to show you two different sorts of tools to visualize that sort of help make sense of how the evidence. So this is SVViz, which is a really useful tool. It shows, because it breaks it into the two alleles. So this is the reference, right? This is that 3.5 kb chunk that is proposed to be deleted, and then this is the alternative. So this is with the deletion. So you can see, like, this is the same thing from the red to here, and then the gray from here with the blue deletion chunk pulled out. And so what's cool is this was they was predicted as a heterozygote, right? So one allele regular and one allele with the deletion. And so this is the reference, and you can see the reads that support the reference. And so it looks pretty normalish, right? Like you have a set of reads, pretty standard coverage across the whole thing, and everything's good. And then you can see the deletion. All these reads are the reads that would look funky on this one are pulled up here, right? And because they all, you can see them spanning so there's two sorts, right? This is the forward and reverse. There's two sorts of evidence. One is these reads that span the breakpoint. If you try to map them to here, they don't map right? This maps, it's called a split read. You have this part mapping way over here, and this part mapping way over here. And so that's all evidence there's something wrong there. And then you'll also have the pairs with each other, and the distribution of the pairs will be different than you expect, right? They're closer to each other than you expect. And so those two sets of evidence are what led the callers to call this. And then you can and this is a nice tool, I think, to then visually break down, where everything is and how they lay out. And then, then there's other tools as well, which I think give useful complementary views of the data. So this is called SVPlotIt, which also, this is that 3.5 kb deletion, and it's showing, it doesn't show alternative, it shows the code of calls there where they're calling at, and then what the distribution of reads look like. So this is more like what you would see if you had like a read depth-based caller, like CNV kit. You have this coverage and it inserts and everything's good, and then all of a sudden, whoa, something's going on there, and then okay, cool, back to normal, right? So the insertions get crazy, and the depth disappears into the thing. So it's kind of another set of evidence that, hey, this is the sort of thing you do if you see the call and you look at it and you say, hey, this call looks real, of interest. So hopefully you believe me that this is a real call. What do you think? Everyone's on top of it, yeah. So it's in the VCF, it's in there, and then you can start to then take this sort of information and then look at what's the context of it, where does it fall, and so it falls in this gene KLK15, which I knew nothing about before I put together this n=1 example, but I'm doing a little like amateur sciencing here, right? So you can look at it in UCC and see where the event happens at, get sort of the context, and then you can do more amateur sleuthing by looking at Wikipedia about what the heck KLK15 is, right? And it's a protease, and there's some association with cancer, prostate cancer, I think specifically, and so it's kind of interesting in that, like, it's a diagnostic for
potential diagnostic for prostate cancer. And so because these are our personal genome project patients, it's kind of interesting that you can then take this and look back at reported symptoms and things and see if there's anything that matches sort of what they're finding in their health records. And one thing that there wasn't anything, he didn't have anything about prostate cancer, but what was interesting is if you look at this is more sort of information, this is GTAX associated information of the of gene expression, and you can see where this gene is expressed. And so there's prostate, which is the one that that they mentioned. You don't hear is testes, which is still probably related to that as well. That's the cancer. That's where it's known as a cancer marker. But then it was interesting. I'll show you why. Thyroid. It's also expressed in thyroid as well, which is not as expected. I think there's a colon over here, so that's probably prostate-y type stuff. But the thyroid was kind of interesting. And he had reported that having thyroid nodules, so some sort of precancerous thingy, right, that got removed in his thyroid from 2006. So kind of this is like N=1, nothing proving anything, right? It's not really science. It's more like a
case study.
Case studying, yes, case studying, exactly. Case studying is a good way to put it. I think sort of demonstrating to me, this demonstrates a little bit of the value that you can get from, I think sometimes with the Personal Genome Project and a lot of these things went through a big hype of like, hey, you should get your genome sequence, you're going to discover all this stuff. And what we really discover is what we already know is that it's super complicated and we don't have any clue about most things. And so you're not going to like discover something magic about yourself, probably, right? Unless you have something wrong. But there's actually a lot of really interesting information in there and useful things that you can, I think sometimes it's useful just to be, if you ever are sick, it's nice to know, hey, this is a potential cause of this, even if there's nothing to do about it, or I don't even know if it's real or that sort of stuff. It's kind of useful to have something. So I thought this was serendipitously things that I found that did not expect. Just basically, I was just trying to put together an example of something interesting in this SCs, and then that's what kind of came out of it. So that's pretty cool. So that's kind of the outputs of sort of the workflow I showed you. So what it tried to demonstrate is you can take this workflow, you can run it in a lot of places, so you can actually do it. You can do it wherever you want, and you get outputs that you can pretty easily sort of query, some tools you can use to do that, and look at that are integrated with bcbio, and then also things you can do on your own. And as a sort of wrap up at the end here, I wanted to talk about automated validation and making sure this works in multiple places and that kind of stuff. But before I go on, are there questions, thoughts about that? Anything people want to?
So I understand.
Yep.
So bcbio is installed on each of these platforms, right?
That's right. Yeah.
Is the installation always going to be identical? Like, so if I take a CWL file and run it in any of them, will I always get the same results? Or can people configure it so it's installed a bit differently?
Right. So, I mean, as long as you're using, if you're using containers, then the answer is it's the same as long as you use the same container. So there's a bcbio will it essentially has a CI system, like Travis CI, that builds the Docker containers at sort of regular intervals, and so you can take any sort of snapshot of those over time and use it. And so as long as you're using the same one, you would get identical results across any platform. That's right. And then you can update or change over time as you want, and you'll get different results when you change it, right? Or potentially different results. And the validations are kind of the way that are meant to say, how will the results change if I do something different? Because the useful way I like to think about reproducibility is that, there's two ways, right? One is like I'm running the exact same tools on the exact same thing, and so I'm gonna hopefully get the same result, I think the other way that I like to think about it, which I think helps you evolve a pipeline over time is, as it changes, am I getting functionally equivalent results on a useful set of validation cases? And so that's what bcbio tries to enable, is to say, hey, I don't have to stop because I checked GHK 1.5, 6 years ago, and I'm just going to keep using that forever. I can keep moving forward, but now I have these set of cases, so I can see how it will hopefully impact my samples, and I can have faith, I can have trust that I'm going to also get quality calls as I go forward. So that's a long way of saying you can run the exact same thing. What we try to push for is having things continue to work well and work in a predictable way over time as the pipeline evolves. Cool. Yeah.
How can the Gemini database be used for annotating, say, the dog variants?
Yep. For dog, I mean, so Gemini can be used for anything as long as you have the inputs. Yeah. So the advantage of human in this case is that there's a lot of pre-existing information about it, right? So there's like 1,000 Genomes Project, there's exactly a whole bunch of other like external resources that exist for human. And so if you have equivalent useful resources for dog, it can use those can be included as well. The framework is generic, so it just takes the associated files, puts them in, and then puts them in a database so you can do queries. But you have to like actually have the resources. I just don't know enough about dog to know if there are like there should be, right? Like there's those projects like the canine, the Broad one, right? There's canine something where they're sequencing a whole bunch of different kinds of dogs. So 10,000 canines, I don't know, maybe. But yeah, I mean, like as long as they exist, yeah, 100%. Like it's a general way of representing things. Yes, that's it. That's it. Cool. So that's a nice segue into the validation stuff, and why I think that's important. So to me, validation is one of the most important things that bcbio does, and is one of the reasons that I think moving CWL is really valuable, is that you can spend less time on infrastructure and more time on this sort of building up validation cases, having a lot of validation cases. And what they are essentially pre-built workflows with known outputs. So ways that you can, things that cover hopefully a wide variety of use cases that are going to be similar to what you have, so that you can have some faith that what you're running will work as well. And so you have things like germline calling, which is sort of the most done validation, but there's also somatic calling, low frequency calling, FFP, structural variants. It's meant to be a big collection of diverse workflows. And so I have a lightning talk at FOSC where I talk about this for 5 minutes and sort of show some of these examples. And this is my more sort of extended. But if you don't believe in the importance of validation, here's why I think it's super important. Is number one, I think it's an integration test for the pipeline. Do things actually work? Did you put them together the right way? Did you not mess something up when you copy GHK best practices into your script? And then it also ends up being a really useful way to do unbiased algorithm comparisons. I end up doing a lot of more than I mean to validations and comparisons between different tools because tool authors are very interested in sort of like, how does my tool compare to this other thing? And they often have a setup that's good for their tool and not for other tools. And so this is a way that they're all a good setup for all the tools. And this gives you a baseline for improving methods and making things better. And then I use them as an automated test for platforms, right? So one of the challenges with CWL is now you're making sure it runs well. And when you port it to DNA Access or report it to Arvados, it should run identically there. And so you should have some confidence that the magic that happens to make your workflow run there doesn't change something that affects the results. And so, it's a way to build across all those different platforms. And so the key of all this stuff is reference materials. Is everyone says validations are important. What we don't do a lot of is building up cases of validations that work well. And the Genome and Evolve Consortium is one of the leaders in this, and it's been around for a long time, and they've really made a good segue into validation of germline calling. Germline calling is very, very good because they have a diverse set of samples that people need to do a good job on calling on that with their callers, and so as a result, callers have improved a ton because once you have a large set of truth cases, then you sort of span the cases of what you need to do, and then if you're doing well on those, you're doing well in general, and they've been really good with that. And the GA4GH has picked up a lot of that validation. Cancer calling is much trickier. There's a lot more cases. Things are much harder. There's not as many true sets. Things aren't consented. The ICGC TCGA Dream Challenge has some synthetic samples which are really useful for that. And then there's also sets of other ones that are developing that are coming out that are really useful. And so I'm just going to walk through some examples of what these validations look like in bcbio and why I think they're useful and interesting. But I thought I'd start off with like what does bcbio spit out. So this is an example validation, just to give you a sense. I'll show some graphs like this. So having one example is helpful. And so on the left here is sensitivity. So how well you're detecting variants that are there. And then this is on this side is precision. So how of the things you detect, how often are you detecting something useful, and how often are you detecting something fake? Right, so how specific are your results? And then on the top, it's stratified here by SNPs on the top and indels, and then here are say 3 different methods, 1, 2, 3. And the only sort of I think tricky thing is that lower values are better. So this is sort of a technical thing, but it's useful to think about when you're plotting sensitivity. One of the tricky things is that ideally you have 99% sensitivity or some high sensitivity, right? And so when you compare two things and you compare 99% to 99.1%, and you have your bar graph starting at 0, like you're supposed to, then those don't look about the same when you look at them, right? So you can't tell what's better than the other. They all look pretty much the same. So one nice thing you can do is do the false negative rate, which is 1 minus it, and then you don't have that problem anymore. So now you can start at 0. If your numbers are small, they can be small. It's fine, right? But you can see the differences in the graph as opposed to. So this is 1 minus and 1 minus, and false discovery rate is 1 minus precision. So lower values are better. So that's a validation graph. What they look like. That's not very interesting because it's methods. But here's
those did you make up those numbers, or are those from real?
I made up these numbers, yeah, method 1, 2, and 3. Yeah, they just made some numbers up. But here's some real numbers, so you can see something of use to you. This is, one thing that I think this is a validation of GHK4 when we're working on that. And what we try to do is these are the 3 different genome and bio samples: 24631, 24385, and 12878, and they're one's Han Chinese, one's Ashkenazi Jewish, and one is Caucasian immigrant from Utah. And so you're trying to sort of span the diversity spectrum. And then I've also tried to span the sequencing instruments diversity spectrum as well. So grab, this is a HiC 2500. This is 10x with X10. This is X10 sequence with 10x tags in it. So make it extra tricky. And this is NovaSeq, and then you do the calling with GATK. This is all GATK, and then haplotype caller, and then compare to the true set and see how you're doing. And so you get a sense of like where the precision is and where you have issues. And so like this for instance, GHK had issues on this sample here, which is 24385. You can see like it's got a pretty high false positive rate, and that's the 10x. So that data has these extra tags in it. It's got the tags which help 10x do the magic of associating things together, and it wasn't very good at by default masking those tags. And so there's a I don't have the thing here, but if you go to here, you can see sort of as we walk through, there's a whole analysis there. We walk through sort of, can you trim out those tags without removing the sensitivity and get rid of some of that, the false discovery rate. It turns out, yes, you can, and it can do about as good once you do some trimming upfront of that data. And so it's kind of nice. It's interesting. There's an interesting comparison there as well with like Freebase, which is the 10x, the one 10x recommends for calling, because it does an okay job from the beginning of handling these tags, but you need to do extra work for GHK. So it's a nice example of, I think, the value of this validation, because number one, where it seemed like those GHK4 work, and you're like, yes, it works really well. You can see how good it does on some samples, right? You know, and then you say, it worked fine on NovaSeq, yes, and they were fine on 2500, yeah, but okay, I've got some 10x data. Is it cool just to throw it at the bcbio pipeline? And it turns out, no, from the beginning, it wasn't cool, but we fixed that, and now it is cool. So this is the sort of thing where the value of sort of a community annotation is exactly in this. If you're not doing these validations, you're not doing this, then you essentially just throw data at it, and you get out variance, and you have no clue if it's doing well or not on your specific data type. And so, you know, I think the challenge, and like I'll talk more about some of the examples tomorrow. I'll let you talk if you're interested, and you can see sort of some of the examples of different things we're doing. But what the advantage of this is that now you can start to automate this validation across multiple platforms. So what we really need is this as a trend is a continuous integration process where we can take where we can do this, run all these different validations in multiple places every month and whatever, weeks, 2 weeks, whatever makes sense, right? And then collect the results over time, show the graphs over time, let people see how the tools are changing over time, and that sort of thing. You know, you can add new workflow definitions, you can test tools and algorithms, you can add them into this. And it'd give you this transparent process where instead of the way things happen now, the way things happen now is GHK or whoever releases the tool, they have a paper or a blog post or something that shows how the tool did, and then you go on to the next one and they do the same thing. And there's no easy way to track that over time, keep track of it. And so this is, I think, one of the critical things that hopefully we're moving into with bcbio. And if you're interested in this, Brian O'Connor from UCSC is a big proponent of this and has, I think I have his, yep. So he has a workflow coordinate execution challenge, which finished, but is featuring a lot of these workflows for validation. And the next step is this continuous integration validation setup. And so, you know, if you're interested in this kind of thing, this is a good example of something that that needs people who can code, that like to build this sort of infrastructure. That's something you could reuse in a lot of different places, and it's a great thing that, you know, I think as a community we should be coming together on. And, you know, for the bcbio perspective, is something that now that we have CWL, if someone built a system, or when we build a system, I'll be positive, right? When we build a system that does these automated validations, you just plug it in and it runs there, and you can add new things and contribute to that. And so, you know, that's the sort of next steps with that. If you follow the GA4DH at all, this is a plug for the work they're doing, is the automation. There's a high-level thing called WES, Workflow Execution Service, which you feed things CWL in a standard way, and it runs them. And it's a shared API, and that's something that I know that they're really interested in having contributors for. So if you are interested, if you like to build infrastructure, or you know people that do, that this is a great thing to contribute to. Okay, so that's all my talking. I think I'm going to try to summarize with what we talked about, and then I'm happy to have more questions or about anything. So we talked about sort of motivating for what bcbio does, which is overcoming these barriers to building analysis pipelines. So instead of having to do everything yourself and then maintain it and build it over time, we're doing it together as a community, where we're all working together. And I showed Come Workflow Language how that takes the complexity the incidental complexity that forces us to use different tooling systems and removes that so that we can work together as community, as more of a community. And then talked about sort of how bcbio practically runs with CWL. So I showed an example of that the PGP workflow, that how it ran on Arvados, how it ran on DNA Nexus, how it ran locally with Foil, how it ran on a cluster with Cromwell, all using the exact same setup. And so sort of how you're overcoming the barriers to running things that way. And then I walked through that example of the outputs to sort of show some of the useful small variant and structural variant calls that we have. And finally, I tried to motivate for, I think, where I hope this is going, hopefully, is to be able to set of useful workflows and a set of really useful validations, complex validations that we're maintaining together as a community and then testing and working on over multiple places. Cool. And so, I hope that, the takeaways you get are, number one, what science really is collaboration and reuse, and that's why we're here. That's why I'm so jazzed about the conference. And, this is our first time doing this with Galaxy and BOSS together, and it's awesome how many people are here and doing training and everything. So, I think everyone gets it, right? And bcbio is trying to get it, and with interrupt workflows, hopefully you could take away, hey, this is the way I can run a bcbio analysis where I want it to run. If you hadn't looked at variant calls, hopefully take away some interpreting variant calling, and then I think just hopefully feeling, like I said, ready to smash the wall or whatever, and build some good things together. And we'll be here, Collaboration Fest. Hopefully lots of people will be coming, and we'll be working on bcbio things and some morpho language things and all these sorts of things. So hopefully have people there. And I think that was all my things. So happy to answer more questions or anything. It's okay if I turn the lights because I feel like I'm in the dark. Are there other burning questions we didn't get asked during all that?
Is there any kind of formal work among the community on more like N=1 or clinical kind of direction? In terms of
Like
the pipeline, like getting it into the clinic?
Yeah, I mean, I think the biggest work is really the containerization and Dockerization and Singularity type setup, because I think the one thing that Galaxy accepted, most of the workflow systems are a little bit they're not as strict about what goes in and out, right? And so you don't have like a full traceability of everything that happens. So I think like that's number one, and then number two is the platform sort of that do it. So like I know like Arvados is set up for that sort of system because they have like full provenance and traceability of everything that happens throughout. And DNA Nexus also does that as well, and they're like set up in clinical environments. So I think like the CWL plus the containers in those sorts of workflows is getting there. And then, Galaxy is sort of the open sourcey equivalent of that, which has full traceability as well. So once they support CWL, then you've got it all right. Because I think they're doing good with containers as well. So yeah, I think that's where it's moving. And I mean, in my perspective, I feel like there's not a separation between what we should be doing as a research component and what we should be doing as a clinical component. Like, we should have the same traceability for our research so we can go back and see what happened. And so, like, I think it's more a matter of if you fulfill all those things we should be doing anyways, then you easily fulfill the CAP and the CLIA and the requirements, right? You just have a lot of paperwork to fill out in the clinical environment, but on the technical side, you've got everything locked down. You can trace it back and stuff. So. Yeah.
You just need something that doctors can understand.
Yes. Yeah. And so then the reporting, right, is a big component, right? And so, yeah, I mean, I think like that's hopefully, that's sort of another hidden, well, not hidden, but that's another benefit of CWL is that if you can get this if you're happy with this in your system, then, bcbio could be the engine that runs your variant calling, and then the thing that does your clinical reporting is just another CWL tool that connects to the outputs of that. And fits into your system already, you know, and so you don't have to, not everything has to be inside of bcbio. bcbio is just a tool kit amongst the many things that you use. So that's the other component as well.
Sure.
Your slide about continuous integration,
Yeah.
were you suggesting to use bcbio, like, let's say I was working on this tool, it was part of a workflow, and I made a change, and I want to just use bcbio to just run the workflow, you know, like kind of every time I make a change and check it in, just like the test runs.
Yes.
It's like this, you know, in bcbio helps you like validate that output after the test run.
That's what ideally would happen. I mean, yeah. I mean, some of these tests are longer running, so then it's a question of like how often do you run it, right? Like, is every check-in the right intervals? But I mean, that's another. But like, what I do now is like there's a set of like small CWL tests that run every time bcbio checks in, and then there's a set of validations. But what I like is if those validations happen at some regular interval, right? Like every couple weeks, and yeah, and then it validates all the stuff that's happened up to that point, and you have a known expected outcome and some how much it can vary. You define how much it can vary from that, and you get warnings if it doesn't. You get a have a database of the results over time, so you can see things change, or something you know.
And bcbio has facilities in it to do that testing and that validation thing, or you'd be using other tools and just kind of using bcbio to glue them all together.
Right. So bcbio has the validation. So right now, like all those validation plots that I showed are like would spit out of CWL, feed a bcbio automatically, like if you have the validation input. So it'll do the comparisons to the true set. It'll give you the graphs. It'll give you numbers of the results. What's missing is like a framework that would like then take those outputs and store them in a database or something. Query them over time.
Isn't that kind of what Jenkins is? I mean, you can go back and look through all your old tests if you save them.
Yep. Yeah. Exactly. Yeah. I don't think it's like it doesn't exist. I think it's like someone needs to do. There's a lot of plumbing that needs to happen that someone who is good at that sort of plumbing would do. And so I'm always pushing for this project because it's a very good like. I would like to help with biology, but I don't know anything about science, like, or, then this is like a very like project that would, people who understand Jenkins and the magic could do, right? Whereas I'm on the side of like Jenkins does some magic. I don't know. But yeah, absolutely. Yeah. I think that's exactly right.
Can you talk a little bit about the intersection of CWL and WDL?
Yeah.
Because I've never used WDL. I'm just wondering, what are the trade-offs?
Yeah, so I mean, I think like right now the trade-offs are CWL is easier for like if you were a workflow engineer and you want to implement a workflow system, CWL is easier to implement. It's like built from the ground up to be portable across multiple systems and supported. The trade-off of that is that writing it's like a YAML format. It's not quite as nice. It's not like it's more like writing Ansible scripts if you've done that. And then WDL on the other hand, is one of the strengths is it's built like as a language, so it's nice to write. It's easy to write. The downside is then there's a lot of parts that are sort of Cromwell specific that are not well spec'd out. So if you were writing your own WDL system, you would have to sort of know, it would be harder to know how to do those. And so I think like the work that's happening is the Cromwell team has just started supporting CWL, and so now they have an object model underneath that supports CWL and WDL, so they have those interoperating, and then the open WDL, the part of the idea of that is then to like carve off those edge cases so that you have like a base set of cases and then a set of like supported things that Cromwell does. And there's a traction there because like Cromwell is like and WDL is like what drives Broad's internal work, and so they're fulfilling specific needs there. And then it's backporting those needs, I guess, to be like the community for other people to be able to take up and run WDL as well. So what I think, so there's an intersection there that is like a useful set of functionality and that's portable, and that's what like they're trying to converge on. And so what I hope will happen is that you will be able, the goal, and a lot of the GA4 goals are the same as this, is to be like WDL is like a specific way of writing things, CWL is another way of writing things, and they both can compile into something that you can support in multiple places. But I think right now, like Cromwell is mostly the biggest game in town. I think Toil is working on support for Whittle as well, but if you run on Whittle, and you're happy with that runner, then it's good. If you want to do a lot of the running in lots of places, that's where the CWL is better right now. Yeah.
Is Conway supporting Whittle right now?
Oh yeah, Cromwell. Yeah, Whittle. That's like their baby, and then they were.
So they started with Whittle.
Yes. Yeah.
CWL.
Yeah. So Whittle was like a sort of Whittle was like a language at the Broad to write things. I think that they had some other thing before, and then Cromwell was like the runner that would run those, and I think they saw a lot of value in what's happening in CWL in terms of portability, and now it's a matter of sort of bringing those together. Like it's sort of different. And there was initial discussion about like Whittle being like a domain specific language for writing CWL, but like as things do, like you need to get work done, and so they were getting work done, and CWL is getting work done at the same time, and now they both are actually functioning and doing things that they want to do, and then now bringing them together is sort of, I think, the goals there. Yeah.
What your future plans for CWL? Like now, CWL way from the perspective with some pipelines as we say, and make them more universal to scope. Because I mean, maybe in the future, CWL will go the way it's going right now. It might be more useful to have more CWL files.
That's it, yeah. So I mean that's exactly the direction, yeah. So the goal, I mean, the CWL that's there is becoming more feature complete with what's in sort of regular bcbio, and the runners are getting on par with what bcbio had. So what the goal is to push everything to CWL. So I hope that in a year there would not be another way of running it. Everything would be CWL.
So you will kind of replace this with CWL.
Yep, that's it. Yeah. So the workflows will be CWL, and then bcbio will have no runner. There won't be a runner. It'll just be CWLs that you use in other systems. And yeah.
And in CWL, you can, I mean, there is a lot of dependencies like the size, genome, version, and everything. Would you kind of, I mean, the idea how you will combine it into one package? Because I mean, CWL is like a description of flow, but the input is also important. So if you have, I don't know, specific version of the genome, load it, various workings.
Yeah, I mean, I think that bcbio will still be like a generator of CWL, so it won't be like a set of CWL workflows. It'll be more like you enter your inputs and then it'll generate it. Because there is that, like you said, there's value in that, in like you can talk to the platforms and get where things are at, and then if you can also sort of exclude, and you can be smarter about, this isn't doing structure variant calling. I don't need the structure variant part of the variant calling pipeline. You can have sort of one pipeline and just chunk off the parts you need and put them together. So I think, yeah, rather than like having a handwritten set of descriptions, it'll probably be a generator for a while until like, yeah. But yeah, that would be, but having everything that bcbio does get generated in CWL would probably be the next steps.
Right, is that the interesting? Because so my understanding was bcbio would still take care of the biology of the workflow.
Yep.
And CWL more towards the compute and the way workflow is structured.
That's right. Yeah. So
even eventually as well.
Totally, yeah. I mean, like that, just the work would be then around like, hey, how well does a tool work on? Because there's a lot you can happen under the covers, right? Like, how am I filtering? How am I calling the variant caller? How am I, applying it to a frequency variance? Doing other hard stuff that's like what should be in the work of bcbio. I think in the community is building a test case that demonstrates it doesn't do as good as it should, making it do better in that case, and then having that test case in place. That's like, and that's a ton of work. Like that sort of like those validations will take a long time, and so that's like where I'd like to focus the work. And CWL helps that happen, because then you're not, because now there's like making the runner work, making sure things run, that sort of stuff, and then and then there's a lot of gaps in what bcbio supports, right? Like if you want to run on Amazon, it doesn't run that. It's not. You can run on a single machine or a couple machines, but it doesn't run very efficiently. Like there's a ton of effort with Cromwell and other things as well to make them run nicely on AWS Batch and all the newest stuff, and then you just get that for free with bcbio without the bcbio community itself having to develop it, right? Like you're using that other community that's already building that, and then just working on the biology fund. So I like biology better anyway.
So when you say that bcbio will be a generator for CWL,
Yes.
Does that mean so you input the YAML configuration file and then it'll output the CWL configuration file, and then the whole workflow will run based on CWL?
That's right. Yep. That's exactly it. Yep.
Okay, I thought it's running this way already.
That's what's happening. Yeah, I just mean move. That's exactly what's happening now. I mean moving everything to that. I guess it's sort of we're in this transition phase where it's like sort of half the old way, half the new way, and we're getting to be all the new way. But
So is there an output of the CWL file for the current bcbio run?
yes, yeah. I mean, it'll run so all those things I showed are real. Like they would run now. There's just like some things that bcbio supports that aren't CWL wise yet that working on making that happen. Yeah. Cool. We did it till 11:30. I can't believe it. You guys ready for lunch or any other questions? Cool. Thanks for spending the morning. I appreciate it. Thank you all. And yeah, if you have questions, I'm here a whole week, and we will be working. And I'm looking forward to working with everyone. It's awesome. Thank you guys.